And now we have 30 minutes before us for questions, discussion, comments from the audience, please. And I request all the speakers to kindly join us here on the stage. So yes, who's going to start, please? Yes, Dr. Ashwini Parekh.
My question is for Dr. Bertels. I've been following your research for a number of years now, and you were working on Craterostigma earlier. My question to you would be, you're proposing both the plant as a model system to understand drought-related responses. How would you present the comparative account of the two? Which one do you think is model better than the other for?
In a collaborative approach, we have been able to get the genome sequence because the Craterostigma genome is an octoploid genome, and this is not easily accessible for genome sequencing.
Quick question. So do you have some lead from Craterostigma which you would like to continue and validate in this new plant, or, I mean, what is going to be your strategy?
Yes, of course. In the second part of the talk, I tried to show you some analysis of the regulatory regions in Craterostigma, and we will try to validate this also in the Oropetium. However, that is much easier because we have the sequences available. We don't need to then use genome walking approach, isolate the promoter regions, and so far.
Thank you very much.
However, there are also differences. Sorry.
Thank you.
Yes, sir.
Dorothea?
Yes, sir. Yes, sir.
Where?
Yes, I am Prakash Lokhande from Mahatma Phule Agriculture University, India. My question is, what should be the threshold value of soil moisture potential for cereal and pulses to screen at laboratory level? What should be the threshold value for all?
Someone or 21?
Everyone. Sharp sir and other, they have presented this thing on the water potential, soil water potential. What should be the moderate and severe? That should be the threshold value. Otherwise, beyond that, you go for that.
Paper or?
Yes, and also a Lynch Lab reduced crown root number paper.
Yeah, so actually I think those are two different phenomena. One is a reduced number in terms of perhaps efficiency of root system development. That's Jonathan's, steep, deep and cheap ideas. And so by reducing the number of crown roots that are produced, that would relate to that type of thinking. The other paper that I mentioned, for those who aren't familiar with it, Jose Dinany in Stanford published a paper in PNAS recently, which is really in contrast with what I presented today, was really talking about the arrest of nodal roots that emerge into very dry soil. This is actually a known phenomena in the agronomic literature called rootless corn syndrome. It occurs when the surface soil becomes really excessively dry and or hot and hard. And if that situation persists, it actually causes vulnerability to lodging, and so the plants can just fall over and you can lose the whole crop. In the severe drought in the States in 2012 in the Midwest where I am, there were quite a lot of reports of that actually during that severe drought. It's not a typical drought, it's a really severe drought syndrome as far as I'm aware of it. And so it's the sort of extreme end of the response that I showed. I showed data indicating that, at least in the lines we're looking at, nodal root elongation can continue down to soil water potentials of -2 megapascals and perhaps drier. But if we, in the system I showed you today, that divided root system, if we actually fill the outer container with oven dry soil, as dry as we can get it, then that does induce that rootless corn phenomena, and you actually get arrested nodal root development instead. They're not dead. In fact, if you measure their water, it's very interesting, it's intriguing. If you measure their water potential, they're actually almost as hydrated as the well-watered controls, the actual root tissue itself. But somehow they're sensing that this soil is really dry, and they just arrest instead of trying to grow through it. So does that answer what you were asking for?
You have some more questions, please? Some more questions, please, from the audience, from the younger group? Yes. Probably I'll take two more questions as I see the time is approaching, but I'll take one from here, then you, please, second. Yes, please.
Thank you, Chairman. My question is to Amal, who spoke at the end about the reproductive growth. I'm Sheshai from the Agricultural University in Bangalore. What I wanted to know was you have looked at the reproductive growth. Can you, from your experience, suggest how we can have a little, at least a semi-high throughput mechanism for measuring the spikelet fertilities in rice or some of these crops?
Yeah, this is extremely small thing that you're looking at. I mean, you can't. I mean, there are techniques, for example, intact, where you isolate little tissues. I mean, it depends what you want to do it. Because, for example, if there's an epigenetic state that you want to look at, one could do like a flow.
No, sorry. What I wanted to know was to how we can now, for example, if we have a germplasm or some mapping population, where we look at the spikelet sterilities or spikelet fertilities, how is it that we can measure it? How can we quickly do that? Can we measure the temperature of those spikelets, or can we measure some of those parameters which can be useful?
You mean the experimental setup?
Yes, in some kind of a mechanism by which we phenotype it.
Well, that's something I guess everybody should establish for themselves. I mean, one can do hydroponics if you like, or I mean, I don't know, depends on the system that you look
Do you have any specific point of telling him about this particular method he's talking about? No.
I wouldn't be able to say it unless we talk about a specific model system, because it's an extremely difficult task to establish in a given lab for a given species. So I think that's at least in your level, what you if I understood right, you need to establish the technique. But maybe I was talking about biochemical technique or single cell isolation, but I think that's out of range.
Sure, I think we can have a discussion about it later. Yes, please.
Thank you. My name is Hamidou Falalou, Ikrizat, based in Niamey, Niger. My question goes to Xiong Kang. You mentioned that you observe a kind of accumulation of proline and raffinose under drought stress. My question is, have you looked if this osmolyte, how they are contributing exactly in drought tolerance? Are they contributing to protect, for example, the cell membrane to some damage during drought, or are they contributing in the osmotic adjustment to help the tolerant varieties or genotype to capture more water during drought stress? Thank you.
So for the proline and for the raffinose, mostly it's in the osmotic increase osmolyte, total osmolyte, increase osmotic pressure to continue water uptake and stress. From published literature, it doesn't seem to protect the memory directly, but it could, for example, proline could stabilize protein. Of course, if this memory protein, it could also stabilize protein structure and drought stress. So yeah, that could be very important. We didn't mention also the fructans also could be very important in this process because it's a very highly accumulated sugar under drought stress. So, thank you.
Okay, you have a question, I believe. Yeah, please go ahead.
I have a question for Bob Sharp. So you showed that some of the tips of the nodal roots were at a higher potential than the surrounding soil. But then they should lose water. And do you see it? Does it work like that?
Yeah, so you're referring to sort of hydraulic lift. So yes, and you can see it, as you put it, further back along the root axis. You sort of have an adhering layer of moist soil. But the root tip, of course, is constantly moving through the soil. And at least there isn't enough water efflux from the tip into the soil to actually produce obvious sort of a wetting front as it moves through. It's an interesting concept. There might be a really micro layer of water, but it's not enough to see, and we haven't measured it. And it's one of the things I'd like to talk to you about, actually.
Okay, I think we have come to an end of this session. Sorry, we have no more time for questions, as I see Anu just climbing up the stage for some announcement. But before she does some announcement, takes over, I would like to thank all the audience, and also I would like to thank all the speakers and my co-chair here, Lizhong from China.